Okay, Internet, so let's try this again. So, in addition to the enhancer sequences that we're going to have, we also have to have different activators available in the cell. So hopefully you're able to kind of guess that the cell will have the appropriate activators to match the enhancers that are going to be necessary for production of the gene that it's going to need. Okay, so only when we have the enhancers plus a full set of matching activators will we be able to fully transcribe these genes. So let's look at each example separately. So our liver cells are going to have, again, our red, gray, and yellow enhancer sequences, and then we're going to have available the matching activators. So notice this red kind of Mickey Mouse hat looking thing. Okay, that's going to match. It's going to be a protein that can find and bind to the enhancer sequence. Our gray sort of rabbit head, that's going to be matching our gray enhancer. And then this sort of yellow bear head, Winnie the Pooh head, that's going to be matching this yellow enhancer. Okay, so this will not be what we see on the crystalline gene, but we do see this on the albumin gene. Okay, so albumin gene on the top, crystalline on the bottom. For albumin, everything matches. We're ready to recruit in those mediators, the general transcription factors, RNA polymerase, ready to go. But because we don't have the proper activators present in the cell, they're just not present, we can't have this sort of DNA binding structure that allows us to recruit in these mediators, the general transcription factors. So crystalline is not expressed in your liver cell. Again, even though both genes, albumin and crystalline, are present. Okay, so we're going to make only albumin in our liver cells. Let's now look at the lens cell. So again, these guys are going to have our pink, gray, and orange enhancer sequences that are going to match our pink, gray, and orange activators. So the pool of available activators. Notice we do not have red or yellow activators in this case, which means that we can't recognize these elements in our enhancer for the albumin gene, only the gray one, okay? So that means that in this case, we can't form that binding structure for our albumin gene, so we're not going to express that. Our crystalline, again, we do have the full set of activators here, so we're able to pull in those mediators, we're able to pull in the general transcription factors, and we're ready to go with RNA polymerase. So we're going to have crystalline made in our lens cells, and we're going to have albumin turned off in these cells because we just simply don't have available activators. Now, if you're wondering, how do cells start off with different pools of available activators in the first place? Just hang tight until our next video. We'll be discussing how these sort of differences can be generated very early in development and sort of tip the scale towards certain cell attributes and others. Okay, so if you're wondering that, that's a great question. We are going to talk more about it in the next video for this chapter. So we've talked about now two different levels of gene expression regulation. We've talked about chromatin modification. Okay, so that's the histone acetylation, histomethylation, and DNA methylation, controlling how tight or loose the chromatin will be. And we've also now talked about ways to facilitate transcription on the gene once we have loosened it up at the first stage. Okay, so once the gene has been transcribed, we'll see that there are a few different ways we can continue to regulate how much or how little of this is going to actually be able to result in a product. Okay, so I didn't want to spend a lot of time talking about it again now because we just covered this in chapter 17. But once your RNA is transcribed, this RNA processing sort of step, this is going to also contribute to gene expression. So RNA stability, how long is the RNA around? Once we put this out into the cytosol, if the RNA is stable, we can continue to translate off of it repeatedly and get more protein from it. However, If it's going to be around less time, then we won't get as much protein. We can also do alternative splicing and use that as a regulatory mechanism. So again, this was in chapter 17. So if you don't really remember this, I would definitively recommend just taking a look back at that. So you can determine whether a functional protein is actually going to be produced. And so alternative splicing seemed kind of random when we talked about it before, but remember that they have those specific AG and GU sites, branch point A. The cell knows what it's doing. It's able to regulate which products are going to actually form. So in some conditions, you may form a functional product, and in other times, you may splice out something or allow something to stay in that will cause the product to be non-functional. So you can also regulate it in that way. Again, the cell's going to control this really specifically. So I did want to include a slide for this, step 3, the RNA processing step. But most of the things that we would talk about, we've talked about in chapter 17. So I just want to point your attention back to that if you still need some time to look at that. Okay, so all of that up through step 3 happened inside the nucleus. Okay, so everything's happened in the nucleus up until now. So from this point, between steps 3 and 4, you can consider that we have exported the RNA out into the cytosol. So this is all cytosolic regulation now. So we're going to be controlling and thinking about RNA stability and how much we can actually translate from things. So I'm going to use an example, and I want to be really clear that this will be an example because I don't want you to try and apply the specifics of this to other situations, but just kind of think about, okay, I see how things could potentially be regulated out in the cytosol before they're translated from this example. Okay, so iron import, you know that cells do need iron. Okay, your body needs iron. So we have to have a mechanism to import iron into the cell. So transferrin is the protein. It's a transport protein that's used for iron import. Okay, and think about this again in kind of a relaxed, common sense sort of way. When the cell senses that iron is low, it wants to bring more iron in. So you're trying to maintain a specific concentration of iron within the cell. If that gets low, you want to bring more in. We're going to make more transferrin to help with that. If the iron is high, we don't need as much, we'll kind of shut down some of the transferrin. Okay, so that's the end goal that we're working towards. Okay, so we're going to be regulating this through kind of a mediation of some other components. So let's go ahead and introduce those, and then we'll see some pictures of this on the next slide. So we've got the iron response element. Okay, this is a sequence. The iron response element is a sequence in the mRNA. Okay, more specifically, we'll see this in the untranslated region. Okay, so we talked about that again in chapter 17. The untranslated regions are going to be either that just upstream of the translation start site or just downstream of the stop codon in an RNA. It's the part that, as the name suggested, does not get translated. Okay, so we're going to be looking at that, but this is going to be a sequence. It's part of the RNA. The other thing we have to know about, sorry guys, so the other thing we have to know about is the iron response element binding protein. So hopefully you're just relaxing and letting the English of that tell you what this is. This is a protein that will bind to the iron response element. So the iron response element is part of the RNA. This is a protein that will bind to that. Okay, and so kind of the magic behind this is that whether it binds or not is controlled by whether it's already going to be bound to iron. Okay, so when the iron response element binding protein is not bound to iron, it's happy to hang out with the iron response element. And again, we'll see a visual of this on the next slide if it seems like a little bit much. Okay, but when iron is around and it's bound to our iron response element binding protein, that protein is uninterested in the iron response element. So you can think of sort of the iron response element, that sequence in the RNA, as being kind of a backup date. So you would prefer to kind of hang out with iron, but if iron's not around, the iron response element will do, okay? So that's the setup, but we're gonna see why that has consequences. Okay, so we're gonna start out in a low iron concentration environment. All right, so here's our transferrin RNA. This is the start codon. Everything upstream of that is 5' untranslated region. Here's our stop codon. Everything past that, including the iron response element, is going to be 3' untranslated region. So again, this region will not contribute to what the amino acid sequence is going to look like because we've already gone to the stop codon. But it is going to help us regulate the stability of this RNA. Okay, so here's some ribosomes. They're just doing their job. So don't worry about them right now. Okay, this is, you don't have to look at everything at once. Just ignore them. Let's hang out over here. So here again is that iron response element that we just talked about. This purple guy is the iron response element binding protein, okay, that we talked about. Notice the protein. We can see through it so that we can tell what's going on, but that's what's happening. Ignore these guys down here for a second. Okay, so this is exactly what we thought would happen. There's low iron. The iron response element binding protein would prefer to hang out with iron, but none is here. So backup date, it's going to hang out with the IRE, this sequence here. So it's attached to this RNA. Okay, so the ramifications of that are going to be that nearby this iron response element are some sequences that just by chance are going to be sort of vulnerable to certain types of degradative enzymes. So these enzymes are the enemy. They're the ones they want to actually chew up and tear apart this RNA, but they can't do it wherever they want. They have to do it at a specific sequence, which is not shown in this picture. Okay, so it's not part of the iron response element. It's nearby-ish, but it's not just that on its own. So we're going to have these enzymes that want to get at this RNA, but our active IRE binding protein is going to act as a shield. So the shield will prevent them from getting to it. Okay, so let's think about this. This all makes sense. When iron is low, you want to bring more iron into the cell. To do that, you're going to need more import proteins, more transferrin. So you're going to want to protect this RNA when you want more protein. So we're protecting it because this iron response element binding protein cannot bind to iron. We have low iron concentration. So it hangs out here and shields the RNA from damage. Okay, so this is an interesting example. Let's see what happens when we start to add in some iron. Okay, so in our previous picture, we saw a bunch of ribosomes. They were actively translating. Here, notice iron is high. That jerk, the iron response element binding protein, is now hanging out with iron. Like iron response element was just a backup date all along. Now that we've gotten iron into the cell, it is not interested in hanging out here. And these areas are going to become vulnerable to these degradative enzymes. So that means this piece of RNA will be chewed up and it will no longer be stable. It will get torn apart. So that means that you're going to be able to, again, see this go away. There will be no translation. This looks, when you just look at this picture, you think, oh man, you could still go here. But eventually this whole thing's going to be chewed up and this piece of RNA will no longer exist. Okay, and so just think about this. This does make sense. If you've got enough iron, you don't need to bring any more in. So there's no need to make any more of this RNA. So let it get degraded. So that's kind of attaching it again to biological significance. So you might be thinking, Why would you do this? Why would you go through all the steps of loosening up the chromatin, allowing the DNA to recruit in those general transcription factors, go through all those mediator proteins and the DNA bending, allow for transcription, let it get through processing, throw it out into the cytosol just to let this happen? Why would all this happen? Often the cell regulates this type of RNA stability at this level, even though it's went through all that trouble, because this is faster. It's an easy way to very quickly get rid of something that you don't really need. So you can imagine kind of constantly making the RNA, but when we don't need it, just kind of like getting rid of it. Okay, and so that allows you to respond very quickly to fluctuating concentrations of iron. As you can imagine, your iron might fluctuate some, and you might have to respond to it pretty much continuously. So even though it is energetically expensive to produce all of this only to destroy it, this allows for a quick response. Okay, so the cell has weighed its options evolutionarily and decided, okay, it's worth it more to be able to respond to this quickly than it is to conserve ATP. Okay, so that is another example of kind of regulating RNA stability. Okay, so we have another example that's going to be kind of a completely different thing. But again, think about this. This is specific to our transferrin, but you can kind of get the idea of how these regulatory sequences can contribute to mRNA stability. Okay, so let's look at our next example. So this example is going to be of what we call microRNAs. So microRNAs are going to be very small RNAs. We'll talk about their structure in just a second. But what we're going to be concerned with is how these microRNAs are going to help us regulate how much translation can happen on a regular messenger RNA that's going to produce a protein. So we can actually regulate these post-transcriptionally using these microRNAs. So let's talk a little bit about their structure, and then we'll get into their function. So these microRNAs are very small in the end. So this is a primary microRNA transcript. You can see it looks kind of long. It's not immediately obvious how many nucleotides are in here, but it's a lot. Okay, so this is what would be produced right away. But the mature microRNA that we're going to use is going to be between 21 and 25 nucleotides long. So that's pretty short. Okay. These microRNAs do not code for proteins. They're a class of RNAs called non-coding RNAs, which there are lots of, and we'll talk about more if you take genetics. But for now, these guys are just going to be short RNAs, and their deal is that their sequences, this 21 to 25 nucleotide long sequence, will be complementary matches for our messenger RNA, and that's going to help us get some like an active effect. Okay, so that's kind of the structure. These guys are initially produced, they're transcribed in the genome. They have a gene just like everything else. And this primary structure, most importantly, is going to form these sort of folded back regions called hairpin loops. Okay, so hairpin loops are formed when the sequences of these two arms that fold back on each other can base pair. Okay, so when they are complementary and can base pair the way that a double helix for DNA would. So we're going to worry about these hairpin loops. They're going to be particularly important because one of these loop structures will be used in the next step. The rest we don't necessarily need right now. Okay, so let's take this guy, just this one hairpin loop. We can kind of mentally discard the rest of this and take it to the next step. Okay, so let's take that guy again. And this is a closer up region. So you can see there's hydrogen bonding happening here. This is complementary. Okay, so note that it's double-stranded for now with its hydrogen bonds. The next step will be to process this. So an enzyme called dicer is going to help process this. And one of our two strands that we're going to be looking at now will become part of the finished complex. Okay, So dicer comes in, notice what happens. It cleaves off this loopy part. It's going to trim up the ends. We're just looking at this orange part right here. So we've trimmed off all of this, trimmed off the loop, okay? And one of these two strands, presumably the orange one, and that's why it's labeled, it's going to become the functional part of this, okay? So the other strand, who cares what happens to it? For now, it's double-stranded. Okay, the fact that it formed this hairpin loop is necessary for dicer to recognize it and for it to be eventually made as part of this protein complex, but we actually only need a single-stranded thing. Okay, so once we're going to have our RNA ready to go, it's 21 to 25 nucleotides long, we take our one strand, and the cell knows which one it wants, and it becomes incorporated into this protein complex. So this protein complex is going to really do the dirty work, okay? So I think about it like this. This protein complex is kind of like a hitman, okay? So it's going to kill the RNA that we're going to target it to, all right? So if you think about a hitman, they don't just randomly kill whoever's walking by. Okay, that's a serial killer. That's a different thing. But they have a target. Someone has to give them a target for them to get ready to destroy it. This microRNA provides the target. So that's the targeting mechanism. The rest of this protein is just the hitman. You give me the target, I'm going to take care of it. Okay, so that's what's going on here. So the sequence specificity will be what gives us the target. So what we're going to see next is that this microRNA will also be complementary to some messenger RNA. Okay, so it's going to be complementary. It's going to be able to base pair. Notice it's just matching this short region. Okay, and it's going to help target this protein complex to this messenger RNA. So we have two scenarios here. Okay, so one of them, notice, it's completely 100% base paired. The other scenario, we may have like some mismatches here that don't exactly pair, but for the most part, we're going to be base pairing. So what happens next depends on whether we're going to have a 100% match or a partial match. Okay, if it's going to be 100% match, this protein complex chews up the messenger RNA. So this red messenger RNA, this is what would have coded for a protein. Goodbye. Okay, so this status right here, this situation is going to make it, you know, challenging, actually impossible to get any protein from this. Okay, whereas if we're going to have a partial match, so we got some mismatches in here, this complex just sits right here, it doesn't leave, it just sort of traps this messenger RNA, and the ribosome has no chance to kind of translate this because it's physically going to block the entire thing. Either way, no messenger RNA can get translated. So this is going to reduce the amount of protein that we can get from this RNA. Okay, so this seems, again, really just convoluted, like why? Okay, but this actually is a very, very common mechanism. It's a lot more common than people thought. Okay, these microRNAs really were kind of more recently figured out or discovered. And people thought, oh, this is probably kind of like a one-off kind of situation. But in actuality, there are quite a few genes that are regulated this way. Okay, so one really, I think, the usual example that people give is looking at flower pigment. So like flowers that are intensely purple and then have like white regions. Those white regions, often what's happened is the purple pigment, the protein has been repressed by these microRNAs. Okay, and these have implications in development. I mean, just all kinds of function. So microRNAs, really important regulatory mechanism. Again, this is all we're going to talk about for cell biology, but if you're really interested in these, which you should be because they do a lot of things, then when we take genetics, if you take genetics, we're going to talk about these a lot more and other types of non-coding RNA. So there's just really a lot to learn there. Okay, so now we've talked about, did we translate it? And remembering, all of this stuff has happened in the cytosol. Okay, so let's say we get all the way to the protein. Okay, we've made it through all these steps. We make it to the protein, we translate it. How can we regulate it at this step? Okay, so there are a few ways this can be accomplished. I'm going to talk about one new one that we haven't talked about yet first, and then kind of come back to circle back around to some things that we have discussed previously, but kind of put them in this context. Okay, so what we can do, yes, we can go to all that trouble to make a protein and then just get rid of it, just like we did the RNA in the previous step. Okay, so there are a couple of ways to get rid of proteins, but I'm going to talk about a really common one. So we make the protein, and then if we want to get rid of it, and again, this is a speed thing, like let's get rid of this quickly, we can mark it to get, be like one of those proteins that's going to be degraded. So I think about this like if you've ever seen Ferngully, which is, for me, that's the traumatic movie of my childhood. When Home Skillet is going around spraying red X's on those trees, that's what is going to happen next. We have to mark this for destruction. So the way that cells do this is by attaching really small proteins called ubiquitin. Okay, so ubiquitin, and this is kind of a 3 strikes you're out sort of a thing. Okay, so 1, 2, 3. Once you get 3 ubiquitins attached, game over. That's when it's time to go. Okay, so when we're ready to get rid of this protein, we just add 3 ubiquitins, and what happens next is this complex called the proteasome is basically going to eat it. So it will destroy these peptide bonds and break this up into peptide fragments. So just break up the protein into small fragments. These can be recycled. The ubiquitin can be recycled too. Go and put it on something else. The proteasome stays around. It can continue to sort of digest and get rid of these proteins. So if we want to get rid of something really quickly, this is the way to do it. But again, with this mechanism, this is for speed. Like, you don't want to start all the way back from the DNA level. We have to get rid of this now, but it's, permanent for this protein. So you only want to do it if you're sure you really want to get rid of everything. Okay, but this is pretty common way to deal with things. But some other things that we've actually talked about before include adding small functional groups or modifications. So we can think about like phosphorylation. How many enzymes in this class have been activated by adding phosphate group, phosphorylation? Like pretty much all of them that we've talked about. Okay, so we know that's a big one. Methyl groups, other small groups, okay, acetyl groups, just all kinds of things you can do to proteins. Okay, you can add just different stuff to them. All right, and you can think about, okay, adding things to them or just keeping them away from certain areas of the cell. So one example I like to use is transcription factors. So those are produced generally in the cytosol, but they're for transcription. Okay, so even the general transcription factors are produced in the cytosol, but they function in the nucleus. Okay, so if you have something that you don't want in the nucleus all the time, you can keep it out of the nucleus until it's time to be used. So you can keep the protein around, but put it away from where it's going to do its activity until you're ready for it. Okay, so that's one way you can regulate them. Okay, and if you're interested in things moving in and out of the nucleus and how it knows, this is a topic we cover in great detail in Advanced Cell. Okay, there are also things like inhibitors, which we've talked about already this semester. Feedback loops, which are just, inhibitors, activators based off of, products that we've already made. Okay, we have activators in this category, small molecules that can non-covalently bind, transiently bind to enzymes or other types of proteins that can modify its activity. So really, there's a lot of ways to adjust activity in the cell. The proteasome is a really dramatic one, like get rid of all of it, but there are a lot of other ways that we can go about this too. Okay. So we've been through several levels of gene expression. We've been all the way through the DNA level, the sort of transcription, will this transcribe level, the RNA processing level, We've then dumped everything out into the cytosol and looked at the stability and some different mechanisms to degrade or otherwise block translation. Okay, we can allow it to be translated, and then we can regulate the protein. So how do we know, looking at a protein, what steps were taken to get it there? It's difficult to know, but the truth is that almost all of the proteins and gene products that we make, almost all of them are going to be regulated in multiple ways. Okay, so we're going to have to like leave the DNA loose, have to be able to get those general transcription factors there. We can then kind of mix and match regulatory mechanisms, but the end goal is to get a specific amount of the product that the cell needs. So in the next video, we're going to be looking at more functional uses for this. Okay, so looking at things like development and cancer, and why being able to regulate things at a very specific level is very important for the cell and its survival.